So let's talk about RNA sequencing or differential gene expression. Differential gene expression analysis is crucial in identifying genes that differ between your treatment and condition analysis. So what is differential gene expression, or where can we get the differential gene expression data? So when we do the RNA-seq analysis or single cell analysis or your microarray data analysis, we get these kind of FPKM values that are fragment per exon based values that we get out of that. So when we get the RNA sequencing data, we use this kind of data to analyze for our RNA based sequencing analysis. So what we do, we get this DESeq data that is widely used, DESeq2 package, which is widely used for this RNA sequencing analysis. So if you want to learn more about RNA sequencing, you can get this, you can get the courses as well. So now we are going to learn step by step how to use the DESeq2 for your differential gene expression analysis. So first of all, you have to get your R and R Studio installed in your computer system. After that, we begin by making sure that DESeq2 is installed. So by that, we just use the install.packages command and then install the DESeq2. After installing the DESeq2, we load the DESeq2 package by using the library word, and then we move on to importing our RNA sequencing data. So I told you earlier that NGS analysis or RNA sequencing gives us some kind of data. So we import the counts data that we get. Counts data is the FPKM values that we get from our samples. So then we import RNA-seq data into our R environment with genes as represented as rows and our samples are represented as column, and then we remove any unwanted columns that are not part of our analysis. Next, we create a DESeq dataset object and then to hold the count data, and then using the relevant metadata to define our experimental and group conditions. So I'm gonna show you that data as well. So moving on to step number 3, exploratory data analysis. So exploratory data analysis helps us understand the structure and quality of data. There are some special steps that are initial and essential steps that are included. First of all, so I'm giving you just a detailed example of how we are going to do. And in the next video, I'm going to go to the code that we are using. And using that code, I'm going to show you how we can analyze that data as well. So first of all, we take the data and import it. After importing, we are going to normalize that data. So we perform the TMM normalization if necessary with WOM function from Lima package, which is the trimmed mean of M values, which is also called data normalization. After that, we evaluate the sample quality using some QC plot and remove the low quality and outliner samples. And then we filter out the low expression and reduce the noise and focus on more significant gene. So moving on to the step number 4, we estimate the size factor and dispersion. So DESeq2 estimate the size factors to account for the sequencing depth, and it is essential to differentiate in sequencing depth and correct the library size. It also models the dispersion, which represent the variance of counts. So by running the commands that we're going to show you, we can also disperse the command as well. So moving on to the step number 5, which is the negative binomial generalization, which is also called differential gene expression and differential expression analysis. We are going to use the negative binomial generalized linear models to determine the differential expression. After that, we are going to fit the model and test for the differential expression using the DESeq function. And then we are going to specify the two groups which are conditioned, and then we are going to compare it with the construct argument in the results. So after that, we are going to move to our visualization section, which is going to be various methods that we can use. For example, we can use the volcano plot, which is also used as mainly used for the analysis. And then we are also going to make a heat map as well. So mainly we are going to create a volcano plot by using the code I'm going to show you in the next tutorial section of DESeq2 analysis.